It is my pleasure to introduce Ruping Sun to our Cancer Center seminar. And today he will be talking about the evolution timing and fitness of a consequence of aneuploidy and the whole genome duplication in cancers. So I will give the brief introduction. He's a graduate from Fudan University, China, and got a PhD. And after that, he did a postdoc at the Max Planck Institute in Germany. And after that, he did another postdoc at Columbia. So basically, he is in the field of patient-derived data and computational analysis of genomic sequences. And our program GM is used to, they call that genetic mechanism of cancer, and this is representing the core of this program. And for me, actually, for example, translational research is very important for Cancer Center, and that is a kind of engine. But this kind of work, or I'd say nowadays, we call that the motor. But this kind of work is battery. So with it, we can, move the motor and move forward. So let's get into his research. And now, Ruping, podium is yours. Thank you.
Thank Professor Mamoto for the kind introduction. Thank you for joining today. And today I want to share with you some of the challenges we face and some of the tools we built to study how copy number alterations in cancer affect the cell fitness using computational approaches. So our lab is very interested in cancer evolution. So we analyze the genomic sequencing data of tumor samples. We reconstruct the history of the genome evolution of these cells, cancer cells. And then we ask the question, okay, can we see anything that is selected during the process of the development of patient tumors? If so, can we pinpoint to certain mechanism? Of course, from the point of the sequencing data. And the reason why we are so interested in this sequencing data is that these data are coming from patient tumors, so meaning that they represent the native environment of the patient tumor development. So we want to basically have this understanding. And today my talk will be divided into 3 parts. In the beginning, I will give you an introduction about aneuploidy. And then I will present a tool that we developed to time when a particular copy number alteration occur during patient tumor development. And in the last part, I will present a new tool that are under development in my lab for inferring the relative fitness of single cells using single cell phylogenetic trees. So I'll first start with the introduction. Oh, by the way, this slide was generated by AI because AI asked me, do you want me to beautify the slide? So I said yes. So that's AI generated. I found it's quite good in making this summary. So aneuploidy, if you search, you will find the definition saying that chromosomal composition that is not a multiple of the normal chromosome composition, which means that basically in the human cell, the number of chromosomes are imbalanced. So in a normal cell, we have 2 copies of each chromosome, and one from paternal, the other from maternal But in a cancer cell, this number is different from 2. And this imbalance of chromosome numbers are usually termed as aneuploidy. But more recently, we extend this definition to not only arm level or large chromosome level, but also to some segmental copy number alterations, which are large enough. And despite the fact that the mechanism generating it may be different. But anyway, the aneuploidy I'm talking about today are the copy number alterations that are DNA, the gains or losses of DNA segments that are quite large. And besides aneuploidy, there is another type of genomic alteration that is prevalent in cancer, which is polyploidy. We usually term it as whole genome duplication. So this polyploidy is defined as the duplication of the entire genome. But if you think about it, we have an imbalanced cancer genome, right? So some chromosome numbers are different from 2, but this duplication means that instead of from 2 copy to 4 copy, we may have like 3 copy, we can have 6 copy, we may have 5, because the cancer chromosomes, they can misaggregate every cell division. So you have, this polyploidy as one of the mechanism or one of the alteration that generates this high-ploidy kind of pattern in the cancer cell. And despite this, these patterns of these changes that we see in the cancer cell, we have very little knowledge about how do they contribute to cancer phenotype. And this is partly because the experimental techniques to evaluate their effect is quite limited, especially in the past decades. But more recently, we have more advanced techniques to investigate them. Sorry. And if you look at, I'll give you an example of this aneuploidy landscape. So on the left side, we see a perfect balanced genome with 2 copies for each chromosome, right? This is, we term it as normal cell. On the right-hand side, you see a cancer cell line, which is a colorectal cancer cell line. And you see the imbalance of chromosome numbers. Let's say chromosome 7, you see 3 copies, right? As well as chromosome 12. But not only this, like, you not only see this like integer copy number changes of entire chromosome, you can also see some highly rearranged chromosomes. These are, we usually term as marker chromosomes in these experiments. And this means that some parts of different chromosomes are joined together to generate a new chromosome. I was puzzled by this because I wonder why this cell can still survive. When I first analyzed cancer sequencing data, and I was asking, how can this be generated, right? And so we know from literature that this aneuploidy can be generated during cell division, where we know this mechanism called chromosomal missegregation. Basically, during the metaphase, if some chromosomes are not placed in the in the same plane as other chromosomes, or it's not attached properly, it can lead to daughter cells that have different composition of the chromosomes. And sometimes this can also generate some pieces left over that will lead to a micronucleus, meaning that that part of chromosome or DNA is wrapped by a separate it's not in the normal chromosome. It's an extra chromosome of DNA that can circularize, which is quite hot recently. This basically is one of the driving mechanism, we think, how aneuploidy was generated in cancer. But speaking of like segmental level copy number change, let's say it's not this copy number, this DNA segment that's changing its copy number is not telomere or centromere bounded. It's in between chromosome arm. This type of copy number alterations are usually generated during DNA repair or replication process. And some specific mechanisms can lead to this, like non-allelic homologous recombination or non-homologous enjoining. Here you can see an example where the unequal crossing over during this non-homologous recombination can lead to duplication or deletion for different sequences. And upon cell division, these two parts can segregate wrongly to different cells. So you can get, for the daughter cell, you can get copy number gains or copy number losses, right? And also another non-homologous recombination pathway is called break-induced repair or replication, where you have the unwanted restart of the replication. So you actually attach to another homologous sequence, then you get a amplification. You get a duplication on that. So these are the mechanisms that we know from molecular studies, how this aneuploidy or copy number change can occur in cells. If we look into cancer genome, okay, this is done by a review paper. We simply calculate the frequency of the gains or losses occurred in patient tumors. Okay, this is summarizing all the public TCGA or ICGC tumor sequencing data. You see that certain chromosomes are very frequently gained, such as 20, the long arm of chromosome 20. And if you compare the frequency of chromosome 20q gain versus the loss of this corresponding chromosome, it's really like the gain is really preferred rather than the loss, because the loss is rarely seen for 20q. And we also see 17p is frequently lost, where we know a classical tumor suppressor gene TP53 is located in this arm. And this chromosome arm 17p is rarely gained, okay? If you look into certain specific tumor type, let's say breast cancer, you can see that from the panel B here, you can see that chromosome 1q is quite frequently gained. Chromosome 17q is frequently lost. And this tissue type specific aneuploidy patterns are well known, and it is actually quite puzzling. And we saw several recent publications providing some plausible explanation for these tissue-specific aneuploidy patterns, including that one of the paper I believe in Genome Medicine states that the cancer cell is mimicking the expression of normal cells in that microenvironment. Basically, they see some similarity between the aneuploidy pattern and the gene expression in the corresponding normal tissue. So that's why they propose this hypothesis. There are also some other explanations, but this question of why cancer cells show tissue-specific aneuploidy pattern is still I think is still an unanswered question. And given this frequency, highly prevalent, highly frequent copy number alterations in cancer, researchers had long thought for a long time, how do we, let's say, identify certain chromosome changes that are recurrent between patients, among patients. The rationale is that if, let's say, a chromosomal change, either gain or losses are beneficial for tumor or selected for tumor growth, it could be recurrent among patients. Okay, so it's more, so assuming this mixed segregation or this DNA repair errors, they're random or more or less random, those things that are really selected by tumor cells or by the microenvironment where the tumor cell resides could be more recurrent, right? Or recurrently see to multiple patients, assuming each tumor is a random trial of this process of the tumor. So that's why they're large cohort-based statistic techniques where we look for regions that have highly recurrent gains, okay, such as chromosome AQ, or if we summarize the frequency of copy number gains, of course, or if we summarize the frequency of copy number loss, we can see certain chromosomes, they may have some higher level of losses, such as here, the chromosome 5. Okay, this is just an exemplified figure. It's not from real data. So, so this frequency-based investigation of copy number changes dominates the early phases of computational studies. But there's some difficulties in this type of research because we have to make a strong assumption that copy number change that are selected in cancer, they are the same. We have to make this assumption. So they are the same between patients, right? And also, this kind of statistics is affected by the rate of copy number change itself. Let's say certain chromosome region, they are more, let's say, have higher rate of missegregation. You may see more, have a higher chance to see changes in that region. So this method will be affected by that. And in addition to that, we know in certain cancer, like osteosarcoma, ovarian cancer, non-small cell lung cancer, or small cell lung cancer, for example, or pancreatic cancer, this aneuploidy imbalance is really crazy. So you see many different complex copy number changes. If you try to summarize this and provide this kind of a table, the signal can be quite hard to identify. So that's why we are not very happy with this method, if just by using that alone. More recently, researchers started to look into other evidences that may suggest the selective effect of certain copy number changes. For example, by this paper by Ramin's group from Harvard, they looked at the breakpoints, breakpoints of copy number changes. More specifically, they look for copy number changes that starts with one end from a chromosome off, either from the telomere or from the centromere. And then they check what is the density of the other breakpoint across a large number of patients, okay? So their rationale is that the other end, let's just look at this telomere bounded chromosome changes. Because it starts with telomere, right? The other end, they can end, in theory, they can end anywhere in this arm, okay? And then they look at the background of this signal, where this end, and they found it's kind of like uniform Okay, if you normalize this chromosome lenses, different chromosome, you find it's quite flat. Okay, this point can end anywhere basically. They argue that if there is a driver, let's say on this chromosome arm, so let's say you can have deletion, of course, from the telomere to here. There is a driver here. Okay, that's the you can have deletion here. But this part if you don't delete this, there's no finish effect, so you can delete. But it's not selected. Okay, but if there's a driver here, upon deletion, the cell will acquire better competitive situation. Then the other end of the deletion will be preferred to locate here. So once you delete this part, where such that this segment encompasses this driver, this deletion will be preferred by the cancer cell. So you will see a lot of breakpoints, go beyond that point. So you will, in other words, you will see a jump of the density of the breakpoints. Okay, and similarly, you can also identify some pattern where there's a negative selection. So in the beginning, if you start from telomere, if you do deletion, because there is no, there is selection here, okay, or some default effect of that. And if there is a negatively selected driver here, meaning that if that's deleted, then the cell is not doing good. Then, of course, the cancer cell will devoid that particular deletion. So this breakpoint density will drop. So this is a very nice concept, how we can use the sequence breakpoint to study this potential location where there might be some selected events. And this paper actually suggests that the aneuploidy or copy number changes in cancer, they are not random. So indeed they have some effects on the cancer cell. We are thinking about from another angle. We're thinking more from the evolution of a single patient tumor. Well, we know that in earlier studies of cancer evolution, for example, this is a colorectal cancer evolution. It's a conceptual cartoon. We know that there are certain driver gene mutations, and each driver gene may empower clonal expansion. And there's this nice model of sequential acquisition of these drivers. And we want to study if the aneuploidy evolution shows similar pattern of this, or can we look into the history of aneuploidy development in cancer patients and get a better idea. So we decided to go to this aneuploidy evolution direction. And when we analyze patient, copy number landscape from a bunch of different tumor types, we found that these tumor genomes are highly rearranged, and most of the cases have high ploidy. These colors just indicates that the copy number is above 2, and the darker the color to the red, the higher the copy number. And we also see extensive loss of heterozygosity in these tumor genomes, which are quite prevalent in cancer cells. We also see this recurrent copy number changes in certain tumor types, like chromosome 8. The long arm is frequently gained in osteosarcoma and breast cancer. Chromosome 1q is frequently gained in breast cancer, and so on. So here we ask a question. Okay, for a single copy number segment in the genome, can we tell when do they arrive during the development of the patient tumor? To do so, we look at the timeline from the germline cell to the first tumor cell. Okay, the first tumor cell should have, let's say, should have acquired the most of the copy number changes, okay, observed in a single patient. Let's say here we have a patient here. Let's look at this one. Okay, here we have a patient, and this patient, you can see that these are the samples from the same patient tumor. They largely maintain the same chromosomal imbalance pattern, right? Most of the samples, they were quite similar. These common copy number changes, they occur in the first tumor cell, okay? So we basically ask, okay, what's the timeline that, you know, when do these copy number change along the timeline to that first tumor cell? And we define two time frames, two time durations. The first is the initiation time, which refers to when do the first copy number change occur on that particular segment. Maybe I should explain more. So for a single segment, you can have copy number 4, 5, 6. So if you think about it, there's a process to lead to that state. It's not like this segment can jump from 2 to 6, right? So there is a step. So we ask, okay, when does the first copy number change occur in that segment, the first one? And then we also ask, what's the time when this copy number 6 or 5 established? Okay, before the first tumor cell appear. So we can divide the time into this fraction. And then we also had some hypotheses here. We hypothesized this aneuploidy occurring early or close to clonal expansion can be identified. Of course, that means we can identify this two time frame, time duration. We also hypothesized that aneuploidies with recurrent timing, let's say, we found a chromosome change that preferred to be gained very early in many patients, not just in one patient. That's likely suggesting some fitness effect. This insight, this hypothesis, is actually from our computation modeling. We have a model that suggests that high fitness ones will dominate the routes or the development of that. Yeah. So we look into, let's say, we look at a particular genomic region. This is simply how we detect copy number. We have the log read ratio and B allele frequency. We can detect, okay, this region has 6 copies. Then we collect the point mutations, somatic point mutations located in this region. And we have a model of how point mutation occur on this region. To make it simple, if you think about copy number change and point mutation, so the point mutation occur before a copy number change or copy number gain. Let's say you have a DNA segment, right? This segment is not gained, but it has some point mutations. If that copy gets gained, the point mutation will also be doubled because you have the same point mutation on two alleles. But the point mutation occur after the gain. They will remain on the single allele. So this actually is quite universal to, this rule is quite universal in this context. So we can build a quantitative model. We can use linear programming to study what's the time frame for each copy number segments to develop. So we basically turn this point mutation frequency data into an estimate of the copy number timing. Okay, so we study this two time frame as I said, the initiation time when the first copy number can occur, and arrival time when the copy number states established. And when we look at by using this tool, we analyzed this tumor that I showed before. For example, this colorectal cancer tumor, we see very early gains. So first of all, for this chromosome 5, the long arm, the copy number states it's 3, 0. It's 3 copy, 0 minor allele, meaning that there's a early loss. The remaining allele got 3 copies. And we detected there's a very early gain on this loss of heterozygous region. And this region encompasses APC. And APC is indeed mutated in that area. And in addition to that, we also see chromosome 2 has a very late arrival time. That means this chromosome 2 established its final copy number. Here, I think it's 4 copy, right before the founder cell established. From here, the tumor starts to grow, basically. And also we see this synchronized, or in a very narrow time window here, you see synchronized gains. So you have a lot of gains that appear in this time frame. This we believe is associated with so-called whole genome duplication. Okay. And if we plot the cumulative distribution of these copy number gains, we found that this genome duplication, you can see a jump basically here, and they very close to the founder cell establishment. There's another punctuated jump here. So we analyze around 150 tumor genomes, and we notice there are 2 types of patterns in terms of the timing of whole genome duplication. So in adult cancer, such as breast cancer, prostate cancer, we found that the whole genome duplication is usually quite close to the founder cell. Of course, they are truncal, so they're universal in the tumors, but they're kind of like very close to the establishment of that tumor cell. So it's not like very, very early. However, in osteosarcoma, which is a pediatric cancer, a bone cancer, the whole genome duplication very frequently occur in between this timeline, meaning that after whole genome duplication, there's a protracted time until the first tumor cell established. That means the whole genome duplication occurred, but the cell still doesn't, it's not cancer yet. So there's a waiting time until the cell starts to proliferate extensively. So these two, of course, it's not that, it's not 0 or 1, this kind of distinction, but indeed, you can see that from the cumulative distribution, the breast cancer, you have tendency to occur, this large jump tend to occur close to 1, right? Here, this jump is close to in the middle point. So this is quite interesting. And we then looked into, we have this timing estimate for each genomic region in terms of their copy number gains. We then summarize for each tumor type. We want to see if there are any genomic regions or chromosome segments that are preferentially late or preferentially early during this development. And indeed, we found that chromosome 8 long arm is very frequently gained close to the establishment of the tumor cell in osteosarcoma. And in addition, we also found chromosome 1, the long arm, it's very early, so it's frequently early in these patients we analyze. So we suspected that this 1Q is probably providing some finished benefit for breast cancer cell. And indeed, our collaborator, they also independently, they actually found that 1Q trisomy in their cell culture system provide, so they call it addiction point, like this trisomy of 1q is kind of preferred for that cell line. And we got this, from this computation analysis, we turned the point mutation information, right, and copy number into a timing information for copy number change. And you may wonder, what does it tell us in addition to the copy number itself? Because when we analyze copy number data, we usually just focus on copy number itself. Maybe that's already enough. Right? So we ask if we can provide any further insights using this timing information. We plotted the timing information we derived against the copy number information itself. And if we look at the right-hand side plot, you see a negative trend. So the y-axis is the timing from early to late. That's the initiation time for breast cancer. And the x-axis is the copy number itself. From this negative trend, we can say that the higher the copy number of a segment, the earlier the initiation time, which makes sense because for a single genomic region, if it ends up with a higher copy number, it must start early, right? So it probably starts early. So that actually makes sense. But it's not, the timing information actually tells you more than that, because if you look at this here, if you compare the orange dots versus the purple dots, you found that the chromosome 8, which is represented by orange, they have the highest copy number. Okay, but chromosome 1, which is represented the chromosome 1, which is represented by the purple circles, they are not the highest copy number, but they are the earliest to begin. So that means this timing information actually, help us to pinpoint what are the likely outliers in terms of copy number changes. This actually is quite encouraging. In addition to that, from this timing analysis, we also found that certain chromosomal regions, I'm just showing you a conceptual example here. Let's say we have a tumor which has whole genome duplication, which means it has high ploidy. We can detect the timing of whole genome duplication here as a clustered gains. In addition to that, in certain cases, we can observe genomic regions that shows this kind of like very long evolution. Okay, this starts here, the first gain, and this is the last, the establishment of that. And this, we termed it as the recurrent timing aneuploidy gains. And this, we suspect or we hypothesize segments with such pattern could suggest these gains are repetitively acquired during this patient tumor development, which either suggests that there's a high mutability, let's say this region tend to gain, or it's actually the cells like it. So that's why the cell keep getting it. Okay. But of course, this only provides some candidate regions for pinpointing which segments are important. So to turn this timing information to some fitness effect, we actually did extensive simulation studies and using both branching process model, where we basically say a cell can divide or die based on certain parameters. And the cell can also mutate to another type, which has a different birth rate. And in such model, we actually can see that the timing is actually has some dependencies on this fitness effect. I'm not going to talk too much about this, but basically, we think the timing we detect actually is reflecting the potential effect of a certain chromosomal regions. upon alteration. And remember, I mentioned in this timing analysis, we found two contrasting patterns for the whole genome duplication in terms of the timing. One is like very close to the founding cell for the first tumor cell. The other pattern is like it's like mid-truncal. It's not close to the founding cell, but there's a prolonged time frame after the whole genome duplication until the first tumor cell is established. And we actually use this mass model to study what's the likely mechanism explaining this. We can see that if the whole genome duplication has a higher fitness, okay, if the whole genome duplication already has a very high fitness, then it's very likely you will see this. You will see this kind of pattern. On the other hand, if the whole genome duplication is not sufficient to trigger the outgrowth, there will be a waiting time, which depends on the rate of other drivers post whole genome duplication. So after whole genome duplication, there is a rate of driver alterations as well. If that's pretty low, that rate of you will wait for a long time, and then you once that acquired, then you will see a start. So this actually gives us some mechanistic understanding of this pattern, but we're still very perplexed by this pattern because when I talk to colleagues, some colleagues believe that whole genome duplication is beneficial, is selected, but some other colleagues think they're not. So it's quite perplexed. So we wanted to study this fitness effect of whole genome duplication in more detail. So we turned into another data type. So we look into single cell data, and we know that more recently we have the single cell whole genome sequencing protocol that are people keep optimizing. Right now we have actually PCR-free protocols actually, and from earlier data we have analyzed some earlier data. These are still based on whole genome amplification. We look at the shape of the single cell trees from breast cancer. We actually found the trees are pretty unbalanced. So that means that the cell, they have different growth rates basically, or you have some sampling bias, but we cannot tell from that data because you can see that tree is quite, the quality is not very good because the cells are not very, the difference between the cells in terms of their genomic landscape are not very large. But we know from our theoretical investigation or simulation or mass model that the tree of the single cells should reflect, or the information in the tree should tell us about whether a certain group or certain clade is under selection or not. Because basically, if you have a cell that has a high fitness, it should proliferate more successfully, such that you have more sample cells from that clade. And usually you also see, if there's a driver, usually you see a prolonged branch, because that means at this point, the fitness is not very good, because rarely sampled, because you rarely branch. At a certain point, it suddenly branch. So you should be able to tell if certain clades are under selection from this topology of the tree. But the tree topology is quite information rich. So you cannot simply look at this branch length and compare this branch length, or because this tree is, they interact with each other. And what I mean is, like, because you sample a bunch of cells from the tumor, they somehow they're intercorrelated in terms of these parameters, including fitness and mutation rates. So we want to have a way to extract such information from the tree. So we developed a tool called Lee-Frank. We model the tumor growth as a branching process model. that I just mentioned, we have several parameters, which we term as model configuration. We have birth rates, death rates for different types of cells. We have the passenger mutation rate, driver mutation rate, okay? And this M simply means how many types you have in this model. Then we can use a tree that's generated by single cell sequencing. We can either use mutation-based trees or copy number-based trees. Both are quite abundant recently. And for our purpose, we are very interested in copy number trees because we want to study the fitness effect of copy number alterations. And by using these two inputs, we can go to the tree. We can go to, let's say we go to this branch, okay? Then we simply build a probabilistic framework. We ask, starting from this point, what's the probability you see that there is only one branch to the leaf and there's no other descendants? What's the probability of that? You can simply calculate that based on this model, because this model actually has a very nice mass property. You can use a differential equation to calculate that. And similarly, at a branching point, okay, at a branching point, you can actually also calculate what's the probability that this cell branch into two immediate daughter cells with two different types or two same types. So you can express it in a mass form very clearly. And then you can also propagate this probability along the tree. So let's say using the probability you calculated for this branching as a starting value, you can also calculate the density along this branch. So in other words, the probability density of the tree can be calculated from the topology and the branch time or branch length. In this, like we call it message passing. We can decompose this calculation and make it super fast. So in the end, we can get this probability of the distribution of the fitness levels for a leaf. So in other words, for a single leaf, you need to consider all the other branches, all the other cells. Then you actually get a value for that. So because this fitness is dependent on all this information in the tree. And this actually turned out to be quite accurate. So we have simulation studies from both non-spatial model and spatial simulation. In this model, we know exactly the fitness of the cell. We know exactly the history of the growth. So we have a real tree, okay? We can also reconstruct the tree based on the mutations we simulate in a tumor. And we found that when compared to these ground truths we know, we have very good performance. So this, in the left-hand side, you see actually the true fitness that in the model. On the right-hand side, you see the prediction from our leaf rank tool. You can see that the high fitness groups are accurately predicted, okay, with the ranking correlation above 0.85. And let's say in the worst scenario, you have a tumor, there's no selection. Tumor, every cell is equally fit, which we usually term as a neutral evolution, which was quite popular in 2017 to 2019. There are several papers about neutral evolution. Even in that case, you have a tumor that's completely flat in the fitness sense. We can also recover that because this information is basically embedded in the tree. And more importantly, we found that in our simulation, this ranking stability, let's say if we have a bunch of cells sampled and we can subsample the cells, let's say we have 1,000 cells, we can subsample the 700 cells. We can rebuild another tree. Then we estimate the fitness for each cell again. Then we compare how stable, or evaluate how stable this different resampled tree is in terms of the predicted fitness. And we found that if that's very stable, that means the accuracy, which is on the x-axis, is also high. So if you resample your tree and you generate this ranking of fitness, if that's pretty stable, then that means it's very likely that this fitness is true. So in real patient data, we never have ground truth for this information. So we want to have a way to evaluate that. And then actually we found this leaf rank performs very well on spatial simulation where we trigger some spatial constraints in the tumor, where the spatial constraints will make the cell geographically distinct. So this creates some sort of sampling bias, for example. We found that it's still quite robust, this tool, to that noise. And we also simulated the event of whole genome duplication in our simulation, just to see if we can successfully recover the fitness of the cells in the presence of whole genome duplication. So whole genome duplication, basically, it increases the rate of mutation, because you have, instead of have just one copy, you have two. So you have two DNA to mutate. So you simply have higher rates of accumulation. And indeed, we found it's quite, you can see that in this spatial simulation with genome duplication simulated, the correlation of the true fitness to the true fitness is still very high. It's most of the cases brought above 0.5. So with this tool developed, so we finally can investigate the question of whole genome duplication. And we turned out to, we found a paper where they have ovarian cancer single cell sequencing data with a very nice protocol, which is amplification free. So there's PCR free protocol. And that tree is very informative. You can look at this tree. That tree is quite good, I would say. And then we estimated fitness, okay? And we also plotted the probability that each cell belongs to a certain fitness group. You can certainly see that from this tree, from the bottom to the up part, you can see these fitness are increasing, okay? So initially you have a very low fitness. This cell has a very low fitness, okay? And you have a jump here, then you have another jump here, okay? Important thing is that this jump between here to here is different from here to here, okay? So that actually means this tumor, the ovarian cancer tumor, which actually has whole genome duplication for all the cells. This is a truncal whole genome duplication tumor. This fitness evolution in this tumor is not neutral, so that there's some ongoing selection following the truncal evolution. And this is true for the 10 cases we analyzed with this truncal evolution. And these 10 cases, we all have very high resampling accuracy. You can see here, the resampling accuracy of the fitness are very high. And with this fitness information in hand, we can actually compare the high fitness group, okay, versus the low fitness group. We can simply divide the cells into two groups, okay, one with high fitness, the other with low group. We can compare the copy number landscape between the two groups. Then we can ask, okay, which region is enriched in the high fitness group? Okay, we can simply do that. And we actually found P10 here. You can see the P10 has lower copy number in the high fitness group here than the low fitness group. And this is not like just happening in this patient. This actually happened in multiple patients across the 10. So there are like 4 to 5 patients has P10 chromosome 10q. Lower copy is also associated with higher fitness. This means that in the whole genome duplicated genomic background, the chromosome 10q is better to have a low fitness rather than high fitness, low copy number rather than high, in order for the cell to grow faster. And then we also look into tumors with subclonal whole genome duplication. So in this single cell data, researchers found whole genome duplication can be a subclonal event. In this case, we intentionally choose a case where we have the red clades representing the whole genome duplication. We intentionally picked that because here you can see the whole genome duplication is almost sweeping the population, right? It's dominating in this tree. But the tree still has some diploid cells, which is marked by this green color. But when we predict the fitness using our leaf rank, we found that actually the diploid cell has higher fitness than the whole genome duplicated cells. This is because the pattern I just mentioned. You see a very long branch here, and then at this point, it suddenly grows into a large population. So this indicates there's some driver happening here. And if I go closer in this tree, you can see that inside this branch where you have diploid background, this diploid cell actually develop into another whole genome duplication. It's red, just means whole. And you can see that they are born at the same time, basically. But this population, this diploid population, actually have more abundant descendants than the whole genome duplicated cells. Similarly, for this whole genome duplicated population, you see that these are pretty low in the fitness, and then they have higher here, meaning that really the whole genome duplicated cells they evolve to higher fitness landscape, but they do not. The whole genome duplication itself does not immediately have a high competitive advantage versus their sibling. And we have some controls for the tree branch length distribution, just to make sure that our model fits the data in terms of the distribution of the branch length. Because one of the challenges we know for copy number change is that copy number change, they are not independent of each other. Once you have a copy number change, it changes the thing that you can change. So they are not independent in that sense. So we want to make sure that our approximation of the tree branch length is still valid in this case, and which we found in the real data, it doesn't deviate much from our theory. So in summary, we have developed two tools for studying the aneuploidy fitness. One is from the timing, which analyzes the bulk sequencing data, and the other from the phylogenetic trees of single cells, which utilize the more recent advancements of single cell protocols. And with this, I would like to thank my lab members, especially Zichen and Chenyu, on their thinking on these projects, and also thank collaborators for their helpful suggestions and their insights and their support. And thank the funding agencies who provide the opportunity for us to investigate this. And thank you for your attention as well. I will take maybe.
I have microphones for anyone with a question.
So thanks much. You contrasted the timing of whole genome duplication in osteosarcoma and breast cancer. In the breast cancer case makes sense to me because when that whole genome duplication happens, presumably by then, important tumor suppressors have already been biologically inactivated. But if it happens early, like in osteosarcoma, it's a very strange situation because now you have to inactivate 4 tumor suppressor genes. Possibly. We know in osteosarcoma, almost all of them lack any functional P53 copy. Often the same is true for RB1 and then P10 also. So you can sort of see why if that happens early, there would be this protracted period of where the tumor clone is, attempting to lose all these tumor suppressor genes. Because any idea how that happens? And under that circumstance too, you have to also sort of think that the whole genome duplication did something to the cell to allow it to persist, to evolve for such a long time. So what might that be?
To make sure that I get your question. So you think if the whole genome duplication occur, the tumor suppressor has to be first?
Well, I would think that would more often happen after tumor suppressor inactivation
okay,
rather than before, because if it happens before the tumor suppressors are inactivated, now you have 4 copies of a tumor suppressor, and that's harder to lose 4 than 2, I would think.
I see. So in osteosarcoma, in the case that we analyzed, the TP53, first of all, has, I think most of the cases, they have TP53 inactivation and loss of heterozygosity before the whole genome duplication.
Okay.
So it's already gone in that situation. And another thing that I didn't mention in this slide is that because the pediatric cancer, we have a very actually shorter time scale. So if you think about the time, adolescent stage, as compared to the adult, so we may have some sort of a signal, some amplified signal in that scale when we analyze the timing. For osteosarcoma, the mutation, whether the mutation rate is constant or not, we actually also haven't estimated. So it could be that this time, we estimate there are some errors, or we amplify this pre-long period. It's also possible. And we also know in osteosarcoma, because of this rearrangement of TP53, and actually we also see amplified rearranged TP53. So in this genetic background, it could be that the rate of that chromosomal changes are higher. So all these may contribute to the distinction we see in this pattern, but we are not very clear what are the causes. But even if you look at the same adult tumor, you see this distinction. Some tumors, they do have the late whole genome duplication. And actually in this ovarian cancer, you see some are truncal, some tumors, some are like subclonal, meaning that the whole genome duplication is an ongoing process. So, I think this can explain, but you're right. I think we should analyze the other effects that, whole genome also buffers the deleterious alterations. So you have some other effect that may be needed in this scenario where osteosarcoma, where you have higher rate of disasters. So you need to buffer it earlier. That could be, yeah, but I'm not sure if that's plausible. Yeah.
We'll have one final question here.
So can your model tell the duplication of the gene is coming from the normal chromosome? Is it coming from the actual, actual chromosome? Such as the MEC gene, if you see have the copy number again, is it in the regular chromosome? Is it in the actual chromosome?
In this work, we haven't taken that into account, but we do think that by incorporating some sequence breakpoint information, we can add on some further stratification about whether this gain is involving some extra chromosomal DNA, or that's possible. Yes.
All right, everyone, thanks very much. We are out of time. I'm sure Ruping will continue his conversation with anyone with a question. So let's say thank you.
Thank you. Thank you for coming.